All right, welcome back. Right, we're going to talk about DNA replication. So all cells need to copy their DNA. That's what we mean by replication. And the reasons why cells need to copy their DNA is because that's what growth and repair is. If you've got to think about this process called mitosis, there's our cell with its nucleus. If that cell is to, if you know, the process of growing is when you make more cells. So for this cell to undergo mitosis, it's going to divide into two cells. Well, before it can do that, the DNA inside here has to copy itself. Otherwise, you'd only have half the DNA in each of these daughter cells. So really important that DNA gets copied at every stage of cell division. All right, so how does that happen? Well, we call it semi-conservative replication. So just a reminder of what DNA is. You can see it's double-stranded and it has complementary base pairing taking place. So we've got this one, adenine. Adenine is always pairing up with the purple one here, thymine. And they are connected by a hydrogen bond. So all of those complementary bases are connected by weak hydrogen bonds. And you can see cytosine and guanine are always connected together. So how are we going to copy this? Is it just a case of, there's our DNA, I'm just going to do a simple ladder to represent it. Do we just do a simple equivalent to photocopying it? Do we just somehow just turn it into two? How do we do that? So we're going to go through how it takes place. So this is our original strand, original DNA here, and it looks like a ladder, and those rungs are representing the bases bonded together. So a little red line here is just representing that there is a bond happening there, a hydrogen bond holding those two strands together. In order for these two strands to essentially become four strands, because we want to make two sets of DNA, it's going to unzip, and the unzipping is because of an enzyme called DNA helicase. So we're unzipping or we're breaking the hydrogen bonds between the complementary base pairs. So suddenly now, all of these bases here, all these nucleotides, they're all exposed. So what will happen next, these nucleotides here down the bottom, they are going to bind on to their complementary base. So for example, if this had been A, then this one here is going to be T, and if this had been G, this one would be C. So we can see how they're forming hydrogen bonds with their complementary base pair. And as they do that, it's going to build a strand here as well. These are going to connect. And these connections are happening because of DNA polymerase. So the enzyme DNA polymerase is catalyzing the formation of these phosphodiester bonds. So just if you remember, these backbones to the strands, if I quickly draw the nucleotide again, it's these bonds here that again formed. So we're connecting the strands, making it one big solid strand. So DNA helicase is breaking hydrogen bonds or unzipping the DNA, breaking apart the two strands. The free nucleotides in the nucleus are binding onto their complementary base pair, and the phosphodiester bonds that form the backbone, they're being formed as well with the use of DNA polymerase. And eventually you end up with something that looks like this. The black colour here is representing the original strand. So this is the original. And now we've also got a new strand. So that's one DNA set there, and the same's happened here. So once DNA has copied itself, you're going to end up with half the old, half the original strands, and the other strand is going to be a new one that's formed complementary to it. And that is semi-conservative replication.